So this is really how calcium comes into play. We've talked about how calcium engages with actin, but we haven't looked at how calcium gets released from the sarcoplasmic reticulum. So this is the sequence of events from where we begin with the action potential. This is at the neuromuscular junction where acetylcholine is involved, and we have the excitation of the neurolemma, right, the muscle membrane. And so here we're looking at what happens from that point to calcium engaging with actin to the muscle actually contracting. So that's going to depend on, again, that motor neuron, which is bringing the neural input from the spinal cord and engaging onto the muscle. It's also going to require the calcium that's stored in the sarcoplasmic reticulum in muscle cells to be released out into the cytoplasm. Okay. So the role of the neuromuscular junction is to provide motor neuron innervation from the spinal cord to skeletal muscle. It's going to have a single muscle fiber receiving input from a single motor neuron, right? So we talked about that being a motor unit. Similarly, it's going to have a typical synapse. So we've talked about the events at a regular synapse, action potential on the neuron, calcium being released via exocytosis, and then we see that acetylcholine, or the neurotransmitter in question, being dumped into the junction and then binding onto receptors on the other side. So that same sequence of events happens at the neuromuscular junction, and that was what we looked at in the somatic part of the nervous system. So acetylcholine gets released, it binds to receptors, which are cholinergic receptors on the motor end plate, and then the end plate potential is brought about. That is the specific type of contraction that is found on muscle. Okay, now where does calcium come into play? So calcium is binding onto troponin and shifting tropomyosin. This is what we described earlier. So if there is no calcium present, troponin is going to keep tropomyosin covering up those binding sites, and myosin will not be able to engage with actin. There will be no cross bridges formed, and the muscle will remain in its relaxed state. If calcium is present, again, coming from its storage in the sarcoplasmic reticulum, dumped into the sarcoplasm, which is like the cytosol of the muscle cell, it can bind to troponin, cause that shift of tropomyosin, which is also adherent to troponin, and then expose those binding sites. And then cross bridges can be formed between actin and myosin, and then cross bridge cycling, including the power stroke, can occur, which is what is shortening the sarcomere. So the big picture here is that calcium is required to turn contractions on, and the absence of calcium is what turns contractions off. So let's look at the steps of excitation contraction coupling. Again, how we start with the action potential at the sarcolemma. The action potential propagates through the T-tubules. So we talked about these invaginations, these tunnels that are found throughout the muscle, and their role is allowing the propagation of the action potential deep throughout the muscle very quickly. There's also two receptors that are involved, the DHP receptors, dihydropyridine receptors, as well as the ryanodine receptors. And these two receptors are going to regulate the opening up of calcium channels that are present on the sarcoplasmic reticulum, right? And allowing that calcium to move from storage out into the sarcoplasm. So the DHP and ryanodine receptors are sort of gatekeeping the sarcoplasmic reticulum and saying when that calcium can be dumped into the cytoplasm. So we open up those receptors, open up those channels, and calcium is dumped into the cytosol. Calcium can now bind to troponin, and then it can shift tropomyosin away from those binding sites, and then cross-bridge cycling can occur. So those are the steps between what's happening with the neuron up here at the neuromuscular junction. Right, so here's our motor neuron. Here's the action potential coming down the motor neuron. Here is the acetylcholine, which is a neurotransmitter stored in the vesicles here, being dumped out into the junction, and then it can bind to the cholinergic receptors, that are on the motor end plate, or the neural lemma is another term for the motor end plate. And then that can begin the event that we're going to look at. All right. So after the action potential is initiated by way of acetylcholine binding to the receptors here, then the action potential has to propagate down throughout the muscle. So the T tubules, which are these green tunnels, are a way for that action potential to go from the sarcolemma. Remember, the neuron is only adhering to the outer parts of the muscle. So we need this to spread deep throughout the muscle to get to all the myofibrils very quickly. So the T tubules allow for that action potential to move deep throughout the muscle. And then it's going to trigger the DHP and ryanodine receptors that are present on the sarcoplasmic membrane to open up those channels, and calcium can be dumped from the sarcoplasmic reticulum out into the sarcoplasm. And then that calcium can then engage with troponin. So that calcium, which is in green here, will bind to the troponin complex. After it binds, it's going to shift tropomyosin. So notice tropomyosin is going to expose the binding sites on actin such that myosin can now engage on those binding sites. And then it's going to result in the power stroke, right? That cross-bridge cycling process can occur. And now in order for the muscle contraction to be turned off, this is where calcium must leave the sarcoplasm. So calcium is actively pumped back into the sarcoplasmic reticulum, and that decreases the levels of calcium that are present in the sarcoplasm. And now the tropomyosin can cover up those binding sites because calcium is no longer bound to troponin. Okay, so the important point here to mention is that calcium is super important in regulating whether or not muscle can contract, because in the presence of calcium, which is in this form, troponin has calcium bound to it. And so we can make cross bridges and we can have muscle contract. But if all of that calcium gets pumped back into the SR, we can no longer expose the binding sites on actin. We can no longer make cross bridges, and so the muscle is going to remain relaxed. Okay, so just looking a little bit closer here at the two states of actin. So actin here in its relaxed state is covering up the binding sites. So these are the binding sites where myosin would engage. And tropomyosin is what is doing that covering up. And so tropomyosin being attached to troponin is how calcium can regulate this covering up of those sites. So when we have calcium come in, the actin can go from its relaxed state to the contraction. Because it binds to troponin, the tropomyosin gets dragged away from the binding spots. They're now exposed, and the myosin heads can now engage, and cross-bridge cycling can occur. Right. Here's that second video again. Please look at these. I think they're a great way to simplify all of this and to just illustrate them in a 3D way. So what we've just looked at is called excitation contraction coupling. How do we couple the events that are happening on the muscle membrane, which is the excitation, with the actual contraction of the sarcomere, which is the contraction portion? So how is the excitation of muscle coupled with the release of calcium and then the subsequent contraction? The gating of this is really brought about by these two receptors, the ryanodine receptor and the DHP receptor. So the hydropyridine receptor, which is in blue here, and then the ryanodine receptor are adjoined to each other. So when the action potential moves down the T tubule, it's going to pick up the voltage change in the membrane on the DHP receptor. So the DHP receptor is a voltage sensitive receptor that will detect the change in voltage as the action potential moves down on the T tubule, right? The T tubule is that invagination in the membrane. Because it is also coupled on the other side to the ryanodine receptor, and the ryanodine receptor is also acting as the gateway, the channel that is present on the SR. When that voltage is picked up by the DHP receptor, it's going to trigger the opening up of the ryanodine receptor. So calcium will leave the storage here in the sarcoplasmic reticulum and be dumped out into this smaller space where it can flood the actual sarcoplasm. So this is how that T-tubule can send the action potential down. These two receptors, the DHP will pick up that voltage change. The ryanodine will actually also receive that signal, but open up that sarcoplasmic channel, and then calcium can leave the SR and make its way out into the sarcoplasm. And so calcium is going to induce the closing of this channel, but also the opening of this channel, but also the closing as well. When there's too much calcium flooding the sarcoplasm, that's going to be a trigger to close these channels later on. And that is going to be a protective mechanism of the muscle to prevent overcontraction, right? To prevent hypercontraction of the muscle. So when we have the opening of the channel triggered by that action potential, we're also going to see if there's too much calcium flooding the sarcoplasm, that will be a trigger to subsequently close these channels as well. Okay. And then just going back here to talk about the termination of contraction. So calcium is important to initiate the action, right, the contraction, because it exposes the binding sites. But we need to remove that calcium from the cytosol or the sarcoplasm in order to stop that contraction as well. So in order to relax the muscle, we have to actively remove that calcium. So this is where the calcium ATPase pumps come into play. They are present on the sarcoplasmic reticulum, and they actually pump the calcium actively back into the sarcoplasmic reticulum, which is preventing any further binding onto troponin, and it's going to allow the sarcomere to relax, which will allow the muscle to relax. So this is not an active process. Really important that we distinguish that. The calcium is not going to want to go back into the SR, and so the ATPase, calcium ATPase pump has to actively pump it back into the membrane for relaxation to occur. Okay, I'm going to pause here and see what questions we have. We've got a few minutes left. This is all that we'll go over as far as what will be on your exam. The third learning objective here, which gets into the twitch and the mechanisms of muscle contraction, we will look at that as a part of Unit 4.